Hello everybody, my name is Angel Ortiz, and this summer I participated in the Biology Summer Undergraduate Research Program. I worked in the Luo Lab underneath Chuanyun, and we explored the cellular and molecular mechanisms of teneurins in neural circuit assembly. So to begin, we'll discuss the role of Teneurin M in the Drosophila brain. Teneurins are a type 2 transmembrane protein. They instruct type-specific matching between two different types of neurons, projection neurons and olfactory receptor neurons. As you can see from the figure, projection neurons originate from the center and proceed upwards and to the right, whereas olfactory receptor neurons originate at the bottom and proceed upwards into the glomerulus. These two are matched together by levels of Teneurin M and Teneurin A. So for example, if you look in the bottom right of the diagram, you can see that VA1D and VA1V are linked together. These two are linked together because they have the same level of Teneurin M inside of themselves. On the other hand, DA1 and VA1V are not linked together, and this is due to the fact they have different levels of Teneurin M. The unique thing about Drosophila is that it has one-to-one matching, which is known as specificity, of RNs and PNs. So our methods were ORN-PN overlap from candidate proteins. We identified a list of protein candidates via apex-2 proximity labeling and subsequent mass spectroscopy. We performed an RNAi knockdown for each of these candidates. So an RNAi knockdown is essentially where we disable that protein within the fly, meaning its function is lost. As you can see, we ended up with 4 different types of flies. We had the wild type, we had a fly where we overexpressed Teneurin M within DA1. We had a type where we overexpressed Teneurin M, but we knocked down Teneurin M as well. Lastly, we had one where we overexpressed Teneurin M again in DA1, but we included the protein knockdown. As you can see, the Teneurin M overexpression and the Teneurin M overexpression alongside the protein knockdown resulted in overlap between DA1 and VA1V. This is due to the fact that the two began to share similar levels of Teneurin M. We performed the segmentation via Fiji 3D Object Counter plugin, as well as overlap quantification by Python. So these were our results. After running a single image through our Fiji code, we resulted in 3 different masks as well as a CSV file. The first mask is of the MZ19 projection neuron, the second is of the 47B olfactory receptor neuron, and the third mask is of the common volume between those two. As you can see in the CSV file, I introduced an artificial object within channel 4. This is due to the fact that there was a bug within the batch processing where it would terminate the process when it would receive an image without an object in that channel. Since I fixed that bug, we proceeded onto the batch analysis. As you can see, I analyzed 5 different lines and resulted in this data right here. We calculated the overlap via common volume divided by the channel 4 volume. As you can see, we normalized all of the results into this data frame. This is just a slice of that data frame, it's much bigger, where it's very easy to read and easy to understand, and can be applied onto this box plot. So what does this all mean? As you can see, the overlap for the 5 RNAi codes that were analyzed are statistically significant above the baseline of no overlap. The 66987 code had the most significant result, which makes it an interesting candidate for further review. In the future, we may begin to extend our analysis into multiple protein RNAi knockdowns and see how that affects the interactions with Teneurin M and the overexpression. In fact, we've only analyzed 40 out of the 110 protein candidates. As you can see, this is the data provided by my mentor for the protein candidates that he himself analyzed. The overlap ratio of these candidates extend well above the 0 mark, where you can see the wild type are well into the range of 0. I would like to thank Liqun for his help. I would like to thank Chuanyun for his help as well. And I would like to thank the BSURP team for all of their efforts that they put into this summer. Thank you so much for listening.